CRPN Central, this is Dr. Fox. Thank you for your continued support of the CRPN mission and CRPN Central. If you like this free education, please subscribe to the CRPN Central YouTube channel today and follow us on LinkedIn. If you'd like to join CRPN as a site or a sponsor, contact us today at info@crpaynet.com. Enjoy the show. Don't forget to share your voice for good and positive change in the industry.
Ouch! That really hurts. I'll never get used to needles sticking into my arms. I really don't appreciate being a giant pincushion for this trial. Why does the sponsor need so much of my blood anyway?
Oh, I'm so sorry. We need to collect your body samples to make sure that trial's treatments are working as they should. Not to mention, it helps us to keep an eye on your blood work levels to keep you safe when you take these experimental drugs.
I guess that makes sense. Still, I hope the pain is worth it and that all these labs make a difference for me and for the patients who get this treatment in the future. How do I know that all of my labs will be secure and protected? Last week, you asked me to come back for another blood draw because my samples went bad.
Yeah, we are really sorry about that. And we do want to keep your blood draws at minimum. Unfortunately, the shipping truck exploded. Oh, and your samples were on that truck.
Oh my gosh, I hope the truck driver is okay.
I do too. But I can assure you that we do everything we can to treat your samples with the respect they deserve. We have trained staff, calibrate the equipment, and everything we need to get your samples processed and to the sponsor's labs for analysis. I guess you could say we do everything possible to make sure your samples were not given in vain.
Good one. Thanks for bringing humor to a painful experience.
No problem. I know it's not ideal coming here to get poked, but it's always nice to see how you're doing. Thanks so much for coming in. I will get this back to the lab, get it processed, and coordinate a pickup before the end of the day.
Labs, a common and routine clinical procedure, and a clinical research procedure that is absolutely critical to the success of a clinical trial for approved drugs and therapies. Labs are time-sensitive, volatile, and complete failures if we do not prepare ahead of time and properly train our personnel. And yet, far too often, they are deprioritized, inconsistent, and rushed to a point of absolute disaster. Sponsors, labs are one of your only sources of concrete data throughout the clinical trial. Never underestimate the costs of collecting and processing these labs, and remember that a site can only perform your trial as well as the resources that you give them. Sites. For every successful lab collection scenario, there are 100 ways to fail. Don't underestimate the efforts that are required for successful and quality labs. Be sure that you give yourself sufficient time and resources well before a patient walks into the door. Trust me, one of the worst experiences you have in this industry will be to call your trial participants and ask them to sacrifice more and endure additional pain because their samples were lost during processing. It's an experience that you will never want to relive, and I highly recommend you take every precaution to prevent it for your site, your sponsors, and the relationships that you build with your patients. Welcome to CRPN Central, the official podcast of the Clinical Research Payment Network. I'm your host, Dr. Daniel Fox. CRPN Central discusses the real issues with our clinical research industry to explore and identify mutually beneficial solutions for all of our stakeholders. We work in an industry of data, and one of the most reliable and scientific sources we can collect comes from the lab work and biospecimens that we have in our trials. Labs, however, are often a second thought during site startup, and overlooking their complex logistics and a protocol's unrealistic demands, unfortunately, results in wasted time, money, and unnecessary pain and discomfort to our trial participants. In an industry driven by a growing hunger for samples and data, how may we improve our lab practices for fast, efficient, and productive clinical trial performance? Today, we will discuss labs, or clinical lab work in clinical research, defined as the collection, processing, transport, and analysis of biological specimens to aid in monitoring of trial participant safety and assessing the toxicity and/or efficacy of new treatments and interventions. First, we will speak with a third-generation phlebotomist who has founded her own company to improve lab quality and trial participant convenience. Then, we will work with a physician who is highly experienced in lab processing, managing, and overseeing procedures throughout the clinical trial process. To maintain valid comparisons, both interviews will use the same questions, and my guests did not know the questions before the interviews. We will reconvene after the interviews, compare answers, and perhaps identify best practices to help improve the lab experience for everyone throughout the industry. Our first guest is a third-generation phlebotomist who will obtain the samples that our trials need to be successful. Shanna Kennedy is the founder of Veteran Accuracy Mobile Phlebotomy, or VAMP, a mobile phlebotomy company dedicated to ensuring sites, sponsors, and CROs have reliable access to the phlebotomy services they need to perform successful clinical trials. Shanna is one of the shining stars in the lab management field, and I am so thankful she's taken the time to speak with us today. Please welcome with me Miss Shanna Kennedy. Shanna Kennedy, thank you so much for coming on to CRP Central today.
Thank you so much, Dr. Daniel Fox. I am so excited to be here.
Would you be willing to introduce yourself?
Yes, I'm Shanna Kennedy. Hello. I'm a clinical research professional with a good extensive experience in laboratory management, clinical trial operations, and biospecimen logistics. Throughout my career, I've worked as a clinical research coordinator, a laboratory manager, a study project manager, as well as a site manager. It pretty much gives me a well-rounded perspective on the challenges that research sites face.
As I understand it, you started your own company dedicated to labs?
Yes, I'm the founder of Veteran Accuracy Mobile Phlebotomy, also known as VAMP Labs.
Nice.
I've combined my expertise in phlebotomy and clinical research to create a mobile lab service that pretty much bridges the gaps in specimen collection, study issues that may arise, decreasing the number of deviations that arise because labs is normally the last thing that the site is worried about and is sometimes after activation. In my opinion, labs are the data. We're talking about conducting phase 1 through phase 4 IP trials, and we want the drugs to get to market, but if we don't have the necessary data from our PKs and all the other labs that we need as far as the process and what the body is going through with the drug, I don't think there's much for us to go off of except for regular vitals and things like that. Ultimately, leaving labs at the wayside without having the discussions at the SIVs to see what the site's feasibility is to conduct the study in itself is really putting your site at a disadvantage.
Today's topic is labs, and I know it's a super short word, but it's a super important short word. I'm going to ask you a series of questions, and I've already asked these questions to another individual. After we're done asking you these questions, we're going to take a look at what the other person said. We're going to compare your answers, and we're going to see if we can kind of see an alignment in how we can do best lab practices throughout the industry.
Makes sense. Okay.
Could you walk me through a site's laboratory responsibilities from startup to closeout?
When you are starting up with a study when it comes to lab management, you want to make sure that you have access to a great local lab, first of all, because one, your screening lab's utmost important is going to make sure that you minimize any screen failures, identify the best patients for the study. We also have to address the site's actual feasibility. Some sites look at their ability to just draw the labs as efficient, whereas you still have to worry about collection times, collection processing times, collection storage times, and you want to make sure that your site is actually set up and prepared for the storage requirements after you have collected the specimen. Whether that is dry ice, you send it right out, sub-zero, -40, -80, refrigerators. Ultimately, then we're looking at our transport. These are just some some of the logistics in startup that you want to make sure that you have aligned. Now, when we're looking at study timeline, we often see that sites will begin a study and then they realize that, okay, one, I wasn't aware of certain labs when it's actually in the protocol, right? But you have to make sure you read it like your Bible and pull that laboratory requirement out separate so you can structure that according to these visits, the timelines that the specimens need to be collected. Then we're going to close out, and then we're talking about any specimens that you have stored and the proper way to make sure that these have been stored correctly, they have been documented, labeled, and ready on a manifest to be shipped so they are not lost.
I noticed you said two terms here. You used local labs and then central labs. What's the difference between the two, and why would you use either of them?
A local lab is generally could be a hospital, normally is LabCorp request. This is the type of lab that is literally local to the site. They're able to send off their specimens the same day and possibly get their results back the same day. It helps them with the screening process, identifying any issues, any exclusion criteria that might come up, inclusion criteria as well. It could be hit or miss. What's most important is that they are listed on that 1572. Now, the central lab is normally not designated by your site. That is something that the sponsors already have contracted, and you have to know how to efficiently communicate with your central lab. This is a point where once your specimens leave your location, anything can happen, and you want to make sure that you are documenting that process, tracking the shipping, and knowing the transport time, processing time of your central lab to make sure that you can get that data back in time. Because I've had experiences where some central labs are like, we never received it, and now, of course, that becomes a missed data point, and now it's a deviation, and you know where that
A
leads.
giant mess. And then it's finger pointing because nobody wants to take the blame for that.
Oh, yes. That's something that they love to do. They will blame the laboratory or the lab management for anything. So I always encourage the staff, hey, let's make sure we have our paper recs, or we print out our electronic requisitions. Let's put them into our subjects binders for each and every time point, date point. I want to keep a documented paper trail. We all love electronic filing and things like that, but there is something when it comes to the labs, they tend to just sometimes drop the ball. They say, hey, we never received this specimen. Well, hey, this was picked up, you know?
Mm-hmm.
These kind of things are of the utmost importance because you don't want to have a specimen that is now pretty much a waste and you have to keep bringing your patient back and drawing them. We're here to mitigate all of those things so we don't have those problems.
Proper planning and proper processes are key, especially in that event. Labs are super important, and it seems like there's a ton of detail around the lab. So do you pull that out of the protocol?
Actually, yes. In the protocol, you can find the lab specifications that they came up with. But a lot of times I've noticed in those specifications, there might be something that requires an amendment to the protocol in itself. For instance, they might ask for a certain processing of a sample that just generally does not equate to the sample. It might require a specific tube and not the tube that they have in the protocol. But it does make a big difference because those specifications will determine drug levels or blood levels. So you want to make sure that you can or cannot substitute certain collection tubes and storage. I encourage to contact your CRA to get a standard of what it is that other sites have been doing. I also try to encourage looking into the TMF.
So what about the lab manual?
So our lab manuals, you can pull that from the protocol. They're supposed to supply that to you when you're opening up your site. That way it's also your handheld Bible. This will actually tell you what tubes, what supplies, the requirements, the time points, the processing. They will give you everything that you need as far as labeling, as far as the shipping, when it's going to be due. It's like the protocol separate from the protocols.
So do you ever run into a situation where there might be a discrepancy between the lab manual and the protocol?
Yes, absolutely. There was I believe, one issue where we were coming into the study and we were waiting for an amendment. And the amendment was actually to the lab manual. Basically, what we had prepared originally for our study, we had to go back. And luckily, we had only started screening one patient. I kind of try to say, hey, get your trial one patient out the way to make sure that everything is covered.
If you talk to any very experienced operational person, they're going to say this may be competitive enrollment, but I'd rather handle one deviation on one patient than to go crazy and enroll like 10 patients and then have 10 times the deviation. We've got to make sure that this is right, and then we can scale.
Correct.
What are some of the challenges that our industry faces throughout the lab process?
Overly complex protocols. They are not considering the real world site and patient burden. They're just like, oh, this is how we figured it's supposed to be done, and this is how you're supposed to do it. Meanwhile, once we tried it your way, this is the reason why we're on Amendment 5, because it's just not going to work. We also have a lack of standardization. Because we are able to use LabCorp, Quest, hospital, whatever is local to us, your site can be in Arizona, my site can be here in Florida. We're going to have different standardizations. And then the logistical gaps, specimen collection, the processing, and the transport. They always think about that after the fact. So that's where we have data loss and we have delays.
Clinical protocols are written by scientists, or maybe like someone they're used to working with animals and like, well, these are animal studies, so we can control all of these variables. And then you apply it to the clinical world and it's just gone.
They go home to their families. They have hiccups. They have to call you to say, hey, I can't come in today. You have to know if there's a window of opportunity. True indeed, some of the windows are decent after a certain amount of time that the patient has been on the study. But then when we look at phase 1, day 1, there's no window. You have 0, half an hour, an hour and a half. So you're looking at 30 draws right here that day for that one patient. And if you do have 5, 10 patients starting that phase 1, then you really have to be on your P's and Q's as far as your time points and being able to collect and process and keep going back and forth.
What is your record number of tubes that were required for a single visit for a trial?
I would say the most that I've had in the study included PBMC, so that was like the additional 5 tubes. And I would say maybe around 15 to 20 was the most.
15 to 20 tubes. That's pretty rough on the patient.
You can also advocate that they allow you to use catheters, because when you have patients that require a lot of blood work for the study, and frequently you just cannot continue to beat up the best vein that they have. I've dealt with patients that were on dialysis in the study. Their port is their port. So at the end of the day, now what I need to get is going to be opposite side. It tends to be a lot of scar tissue. And I advocated, hey, let's make sure we put a catheter in here, because these are the same patients that rapidly scar. That means they're going to close up. They're going to close up around the bevel of the needle. So we do want to make sure that the patients as well are as comfortable as they can be and willing to participate in these trials. A lot of patients don't realize the lab requirements of the study because the CRCs or the site itself was not very familiar with the lab manual and the requirement. They're saying, hey, yeah, we'll have certain draws every few weeks, blah, blah, blah. And in all actuality, it's something different. And now the patient wants to withdraw from the study saying, no, I can't do that. I can't come back and forth to you to get my labs done. That's where we come in. We want to make sure that we keep the patients in these studies.
Have you ever run into that situation where you bring a patient in and you just can't get the labs out of them?
Oh, no.
No?
No.
Never run into that one?
No, that is our specialty. We will collect every drop that we need. I guess it's just it's a family thing. I'm a 3rd generation phlebotomist, so.
Oh, wow.
Yeah.
3rd generation. So you will get that sample.
Yeah, I will obtain a sample. I actually have a course for advanced phlebotomy that I have put out on Canvas Infrastructure. I haven't promoted it yet because I also want to bundle that with a clinical research phlebotomy course to get phlebotomists and equipped at least with some 101 information so they can have the tools they need to go and venture into clinical research labs. We need them. them. We're short-staffed in that capacity.
What are some of the current advantages that our industry has developed for our lab practices?
I think we can do better. Site burden is a big thing to me. They really need to try to separate a lot of tasks from the CRC. The CRC can't do everything. Having an outside vendor to assist or that is willing to partner with you that is already trained or can be trained on the protocols that you are working on, I think that would take sites a lot further. As far as the things that we're doing right, there's so much that we can be doing better. I mean, I've seen so many specimens hemolyzed. That's not doing better. Collecting just off the sake of collecting is not doing better. I focus on specimen integrity. So unless you are collecting the specimens properly and the best methods, I'm not saying we're doing anything particularly wrong and we're not doing exceptionally well, just in that middle ground, doing things in a standard and getting them done efficiently so we can move on and we don't miss anything.
What do you find is the highest probability of a deviation?
The most that I've always seen is just missing a specific lab. It might be immunoassay that they miss. It might be a coag that they miss. Those things are just as important as those chemistry labs. Accessing that lab manual and making sure you have a list of all of the potential labs that can be drawn, and making sure also that you are paying attention to your schedule of events. When, what day, what visit. And then once you're looking at the visit or you have mapped out your visit plan, you can also say, okay, what time do I need to collect this sample?
Yeah, I know a lot of sites, they'll come in here and they'll say, well, the CRO sent us these lab kits and the lab kits have everything we need for every visit. And then they don't. The CROs are busy too. You can't trust your lab kit. You have to trust the protocol and the lab manual because you are going to be the one.
Absolutely. Absolutely. Every lab kit needs to be inspected. I'm going to say that again. You need to make sure that each vacuum container or storage container is not expired. That tends to be a lot of people's issues too. They are going and they're just grabbing the kit because they didn't prepare the visit ahead of time, or they didn't realize, oh, we didn't have enough. So now we got to pull from supplies that we have or don't have.
Exactly. Make sure you have lab kits maybe even before you schedule a visit with a patient.
Absolutely. You want to keep track of what you have, period, and when they are due to expire. That way you can order from whatever lab, the central lab, local lab, whoever it is that you need to get these supplies from, and make sure that you have them on site. You want to make sure you have the storage capacity for lab supplies.
And if I'm not mistaken, you're not allowed to pull supplies from one trial and use it for the other, right?
Correct. And I'm going to say, I've seen it done. Like, well, well,
robbing Peter to pay Paul over here. I know the CTAs. I know what the contracts say. You're not allowed to cross your supplies in your trials.
Absolutely. There have been instances where we've ordered supplies. We don't know what's going on. When are they coming? Why it's taking so long? Contact the CRA, and the CRA is advocating to pull it from the next kits or an old kit
of the same sponsor.
It's within the same trial. Now, the only issue with that is if you're not aware or knowledgeable of what department of the laboratory that specimen is going to be processed.
How may our industry improve its lab practices for fast, efficient, and productive clinical trial performance?
It's always going to go back to preparation. If you don't know what you need, you don't know what you're missing. Have a run through.
What's a run through?
A run through is basically a mock visit. And that's what I do when I manage the sites. I make sure that I train the staff for the specific visit that's coming up. That way everybody is aware of what is going to be needed. All of that is going to determine your labs and how efficiently your visit is going to run.
From your experience, let's say that you're using Central Lab and you need supplies from the CRO or the sponsor. How long does it take you to get them?
I generally give it at least a 2 to 3 week window. You will be left holding the bag if you think that you're going to get your supplies within a week. Just like you want to have a manifest for your specimens, you want to have something similar to that as far as keeping an eye out for the date that your kits are going to expire. Let's say, for instance, we have some kits that are going to expire next month. We need to already be contacting the sponsor to get those ordered in. I suggest just to establish reading up on all of your supplies. If this whole series is going to be expired, you need to replace this whole series, even if you do not have a patient, because you may not know if you have to go in there for a substitute. You don't know if you're going to have an influx of patients that you've recruited and now they're all on this kit. It's better to have it than to not have it.
If I were to hand you a microphone right now and I asked, what would you like to tell the entire industry about labs and what we can do to make them better, what would you tell them?
If we are addressing like lab related inefficiencies in clinical trials, it's just going to lead to higher data integrity and reduced deviations and just better patient outcomes. That's ultimately the goal, money factors and things like that. But we are here for those patients. We wouldn't be here if it wasn't for the patients at the end of the day. I just want them to know that Vamp Labs has been founded to partner with the research sponsors and CROs and the sites to optimize the specimen collection processes, to get them to understand how they need to be looking at their protocol and separating that lab manual as its own so they can ultimately process those things on their own and get ready for activation.
Labs are important. They're probably one of the most important things that we do in clinical research. And as a result, we need to respect them. We need to plan for them, and we cannot put them on the back burner until the end. If we can get our sites together, if we can plan and prepare and create quality standard to the point where it's something that everyone can agree to, maybe we can make labs a better place and therefore a much more efficient way that we can perform research.
Correct.
Well, Shanna, thank you so much for coming on to CRPN Central today. It was an excellent conversation. I can't wait for the live event to follow.
Oh my God.
We'll get to see everyone.
Thank you so much, Jane, for having me.
I'll talk to you later.
Bye-bye.
All right, bye. Our next guest not only performs and manages labs to perfection, but also feels the therapeutic zen of processing them. Preeti Dhaania is the co-founder and director of site development at Cornerstone Clinical Research. She works with sites to ensure they have everything they need, including lab procedures, to deliver successful clinical trial performance and results. Preeti is an amazing professional in the industry, and I am so thankful she's taken the time to speak with us today. Please welcome with me Dr. Preeti Dhaania. Preeti Dhaania, thank you so much for coming on to CRPN Central today.
Thank you, Dr. Fox, for having me. I'm so excited to be on here.
Would you be willing to introduce yourself to the audience?
My name is Preeti. I am a physician that took this new path into research. Research was something that just kind of fell into my lap. I absolutely fell in love with it. It was something that I was good at. I've been in research for a couple of years now, and I ended up founding Cornerstone with my co-founder, Nicole, who was on your podcast a couple of months ago. We ended up making this company just because we were working at a site, a very big site, and we were realizing there were so many things that could have been more efficient, not just from the site point of view, but also from the patient point of view. And we realized we wanted to be able to help others succeed in this field. We basically offer anything a site may need help in, from creating SOPs to running studies itself to training staff. Cornerstone really is there for you at every step of the way.
What does Cornerstone do for the lab systems at sites?
Since I have experience as a coordinator, I processed many labs. I'm going to be the odd one out, and I'm going to say that processing labs was very therapeutic for me. Once you understand how to process labs for a certain study, it kind of becomes very zen. You're pipetting, you're centrifuging, you're packing your samples, and all of that, I feel like in itself is such a great way to calm yourself. I just love labs. I love being able to create a plan, a flow of, okay, I start from here and then I need to do this, I need to do this. For me, my brain loves having plans.
I've heard that from other coordinators where they say in labs, I have these processes. It's like I get to decompress from my study visit. And on top of that, there's timers and you have 5-minute times between what you have to do. And as a result, you really can't do anything else but just the lab.
Yes, exactly.
Today's topic is labs and everything about labs. I'm going to ask you a series of questions today, and I'm going to ask those questions to another individual. We're going to take your answers and we'll compare them to the answers of the other individual so that maybe we can try to find some best practices or try to find some kind of an alignment to how this industry treats labs and maybe how we can improve them.
Sounds great. I can't wait to get started.
Can you walk me through a site's laboratory responsibilities from startup to closeout?
From startup, the first most important thing is to understand if the study protocol calls for central labs and local labs.
And what's the difference?
Well, central lab tends to be the main lab that the sponsor itself uses. Most commonly, it tends to be the lab that's associated with the CRO. And then, of course, some studies call for local labs, and that tends to be tests that maybe they're fine with tasking to someone else. From there, really, it matters on if the local lab is able to process those labs and in what time, because you need results in a timely manner. The local lab cannot take a week to get you a result.
The whole objective of lab is to collect some kind of a biospecimen from a patient and to run a test on it for data.
Yes.
And then the data will be used to maybe make decisions in the clinical trial, or the data will actually be used as a primary endpoint where we can see what the labs are doing. In the world of central labs, it sounds like samples are collected from patients and they're sent to a central location for the performance of the study, for the data generation. And then the data is sent back to the site to make a decision or to put it into the source. But then on a local lab, the data is collected at a local level around the site, not necessarily at a centralized national location.
Yes, that is 100% correct. Before you start the study, you got to make sure you have all the kits you need, you have all the tools that you need. Do you have the correct type of freezer? Do you have the correct temperature monitoring system? You need the correct equipment to make sure that you're doing everything per protocol.
How do you know what equipment you need for a lab?
Well, the lab manual has everything that you need. It's the protocol for the lab. It has the equipment, the tubes you're going to need, the process of which you're going to process everything, how you need to ship, what labels you may need, everything.
It seems like a really important document. When do sites normally get their lab manuals?
From my experience, sites tend to get it before their SIV. Before I bring in any patient onto the site, I like to go through and read the lab manual. For example, we're about to do a screening visit. I want to go grab that screening visit kit.
I've heard you say kit a couple times. What exactly is that?
A lot of times in studies, we receive these boxes or bags that have all these different type of tubes that we need to draw blood for. It might end up being tubes, it might end up being swabs, depending on what the study calls for. The labs are very nice and they end up packing it for us with a label, and the label states what visit it's used for.
They go in there and say, okay, you need to take these labs at this visit, so here's all the tubes you need.
Yes, exactly. But what's more is that when you go into the lab manual, they also have it broken down in there too on how to process each and every tube.
Even down to pictures sometimes, right?
Yeah, sometimes if we're lucky. If we have even more luck on our side, they tend to give us a quick guide, which is my favorite. Going back to the prep that I did before bringing the patient in, before I do any visit, I tend to make my own notes on how to process each and every tube or each and every sample that I receive. Once I'm in my Zen, I bring out my notes. I have all of the samples in front of me. I just follow my notes and the lab manual to a T. The tubes have been clotting for the correct amount of time. Do they need to be on ice right now? Because it needs to be a cold centrifuge. I clot, I centrifuge, then I pipette. And then I store them according to their temperature needs. Kind of just going through that whole processing and really keeping an eye on all the times, because having multiple timers is key.
So you're ever in a situation where it's not just you have to process the labs, but you actually have to ship them that day?
Oh, yes, definitely. Ambient labs tend to have to be shipped the same day because you have to remember that they need to be stable when they receive them. You have to have a plan before the visit even starts. Do I need to call UPS or FedEx and schedule an appointment for a pickup? Whatever it may need, you need to have that done prior to starting that visit.
That could probably really be impacted by the time of the visit. Like if a patient comes in late at night or early in the morning
Also, keeping in mind, can you ship on a Friday? Can you not have Friday appointments?
or a holiday.
Yeah, or a holiday. Also, making sure you have all the labels you need for the box that you're shipping. Does it have a Saturday delivery sticker on it? Does it have a dry ice sticker on it?
Oh, let's talk about dry ice. So how does that work?
Dry ice is honestly one of the coolest inventions.
No pun intended.
Yeah, no pun intended. But also a very big pain. If you've worked at a site, you know, in the summer days, especially in the Vegas heat, you know how hard it is to get dry ice. Every single grocery store is basically sold out. Dry ice is this amazing invention that we tend to use when we're shipping frozen samples just to maintain temperature within the box that we're shipping. So obviously your samples before putting them in the container with dry ice, they need to be properly frozen. When I say properly frozen, I don't mean just, like, oh, we put it in the freezer for 15 minutes and it's good to go. No, because the sample may start thawing on the way to the central lab. And we really want to avoid that because then it may not be stable enough to process and get a result from. Do we have enough dry ice on the bottom, on the top? Is it broken properly? And you have to be very safe while using it, making sure you're wearing the proper gloves, making sure that you're protecting yourself.
Having a dry ice protocol of sorts. When you get lab supplies from sponsors, you get the lab kits that you talked about, but then you also get what they call shippers. And that's those giant styrofoam boxes in a cardboard box that you need to ship the labs.
Yes. It's so important to make sure you have the right type of shipper. There's different types. There's the ambient boxes, there's the frozen boxes, but then there's also those combo boxes as well.
Mhm.
And you need to really be well prepared for those because some of those boxes require you to open them up 24 hours before. Just keeping that in mind when you're prepping for your labs, that you're also prepping your shipping box.
And do you know why you have to open up 24 hours before?
I think it's because it has to expand.
It does, but also sometimes when you open it up, there is a gel pack inside that has to be frozen prior to shipment. If you have a gel pack that you have to use, you got to make sure that you open it up and you're prepped and it's in the refrigerator, the freezer, whatever your lab manual tells you to do.
Yeah, for the allotted time, you're right.
A lot of prep. A lot of prep around labs. So much prep.
Exactly. That's why, again, it's so important to prep the day before. You got to make sure that nothing's expired. And lab kits can be really tricky. Sometimes there's only one single tube in that whole kit that's expired.
Mm-hmm.
Understanding what the sponsor and the lab want you to do. Okay, if just that one tube is expired, do you go ahead and throw out the whole kit or can you switch out that one tube? Understanding what their protocols are for the specific situation. Also, sometimes, let's say you're drawing blood and the vacuum is bad on one of those draw tubes.
Yep. You might need a backup lab kit.
Exactly. So just being prepared for all the things that can happen without creating too much waste. I feel like labs are such a key point of research in general. We tend to kind of just go over them really quickly, but there's so much importance to them.
It's literally one of the reasons why we do research. It's the data.
Yes. When you ship them, you have a tracking number that you receive. Just making sure that that is documented correctly. Some sponsors or CROs, they want to have a spreadsheet just to kind of have a way to keep it organized so they can keep an eye on it. And also us as a site keeping an eye on it to make sure that the packet has actually reached. Quick little story. There's been times where packets have just not reached their destination.
Never arrived at the central lab. And so then they're like, what's going on here? Where'd it go?
Exactly. I remember at our site, what had happened was the truck actually exploded.
Oh,
Yeah.
no.
Oh my goodness. The truck actually exploded. So the samples never reached the central lab. And we were all so confused because when we were looking at the tracking, all it said was sample destroyed.
I hate to see that in the source.
What do we do in that situation? I've also had situations where we did same day shipping or one day shipping and the samples didn't reach in that amount of time because of weather constraints or whatever it may be. And that ends up being a deviation on the site.
Yep, they're out a window. This is a finance thing too. Sponsors don't pay for deviations. That's all there is to it. If you sit there saying, well, it's a deviation, well, was it the site's fault or was it the shipper's fault? Now you start pointing fingers.
And then you have to get all that data to prove your point. Okay, get the tracking information, print that page. You know exactly what time you shipped it.
We did our job according to protocol. We did what we were supposed to do. When something went wrong, it was not our fault. And sites are going to have to defend against that.
Exactly. Once it reaches the lab, then it's basically a waiting game for the site. We're waiting for results to come in, and whether that be actual lab values that you're getting, or it might just be a confirmation page saying, hey, we received it, because all of the other lab values are blinded. Just make sure that you are waiting for that paperwork, and then making sure that your PI sees that paperwork as well, because PI oversight is such a big deal, especially when it comes to labs. They need to know that, okay, my patient came in for this visit, this is the results. Do I need to change the dosage of their IP because certain results were a little bit wacky? Or do I need to make this an AE?
Or screen failures are a direct result of lab values, which
Yes.
means that you're waiting to randomize a patient until you get lab values and you make sure everything's okay before the patient joins the trial.
Yes, very important. A lot of inclusion exclusion criteria is based on those first set of labs.
What are some of the challenges that our industry faces throughout the lab process?
The first thing that comes to mind is the fact that, especially after COVID, it was a lot of being out of stock on certain kits.
Absolutely. I've had so many sites, they couldn't enroll patients anymore because they ran out of lab kits.
Even though it's been 5 years since COVID, I feel like there's still certain situations where we're having that trouble. Or what happens is that the labs or the sponsor end up putting limitations.
I agree completely. Global supply chains are taxed and they have actually limited our ability to be productive as sites right now.
100%. One thing I've noticed just recently with a particular study that we were helping with was the lab manual was very confusing. It wasn't clear. If your lab manual is confusing and it's hard to understand, your labs are not going to be processed correctly.
This is a clinical research site. These are clinicians, they are not scientists. And
Yes.
sometimes I feel like those lab manuals you were talking about, they were written by scientists and they make sense on a scientific level, but they were not written for the people to perform these tasks.
Exactly. You have to keep your population in mind when making things. Another situation that we had was the lab queries.
So there's lab queries now.
Yeah, so if we're all used to EDC queries, those are the bane of our existence, but now there's lab queries. Of course, we have a whole process. We make sure that we fill out the lab requisition. It might be an e-requisition because we don't want to send samples that have not been labeled correctly.
I was going to say, so what is in a lab query? But that might be it. Did this ship on time, or what does this look like, or how'd you fill out the label, or something like that?
Some lab queries can be, oh, what patient does this belong to? Even though the label is right there on the actual aliquot or tube, but then, oh, there was no requisition that came with this. What requisition goes with this sample? A lot of times we're answering lab queries like that where there's missing data, even though it's been filled out properly and it's been sent. But sometimes the lab can query because they can't read your handwriting, which is 100% valid because we all have wacky handwriting when we're in a rush, and half the time we're in a rush.
Yeah.
That's why e-recs are so amazing. The fact that you can just type it or use an app, that definitely lessens the query amount. You have to make sure you're looking at your lab portal every day to make sure you're answering those queries on time, because if you wait even like 2 days too late,
Sample could be done. Yeah.
you have to be on top of just checking the portal for results because one, endpoints, two, safety, but also just making sure you're answering all those queries.
Do sites normally try to get a copy of the requisition or take an image or a photo before they ship it? That way they have a backup.
It should be standard that you should have a copy of your requisition that you're shipping out.
What are some of the current advantages that our industry has developed for lab practices?
The e-rec is a big advantage. One, we're kind of moving away from the paper, which is really nice, going on to e-source, e-reg. There's no handwriting issues. It's typed. We can read it all now. It's also the fact that it's digitally stored. You're never going to lose a piece of paper again.
What is the record number of tubes of blood that you've seen collected from a single patient on a single visit?
26.
26 tubes of blood.
Yes.
And that's for all of these different labs.
Yes. 26 at a single time.
I know that there's a big push right now for low volume labs or low volume diagnostics where maybe we could achieve the same amount of data out of 2 tubes of blood instead of 26.
Yes, I have seen that where they're able to collect more data with the lower amount of blood taken. Because initially there were so many tubes because sponsors had thought, okay, you need a tube for this testing, you need a tube for this testing, you need a tube for that testing. But in reality, how much of that blood is actually being used to conduct that test?
And how much is being thrown out?
Exactly. So if you can combine those tests and take the samples from within that tube, that would be great.
Labs, as stringent as they are, I do think that the industry is trying to work toward a level of flexibility so that we can maybe accommodate our patients a little more, maybe not hurt them as much, take a little less blood. I think IRBs are starting to pay attention to this now too, where you sure you need that extra tube of blood out of this patient? I mean, they are a stage 4 cancer patient after all, and they kind of need their blood.
Yes, definitely.
How may our industry improve its lab practices for fast, efficient, and productive clinical trial performance?
From the site, what we can do better is making sure we're better prepared. Are we actually prepping? Are we actually doing the best we can for our patients? Are we actually making sure that our goal is to help the study achieve endpoints? From the lab aspect, it's very important to make sure that when we're packing the lab kits, we are packing them correctly. There's been many times when I've opened up a lab kit and I see that I'm missing tubes. It's the lab's responsibility to make sure they're packing it well. Take that one job off of the site and help them out. As for the sponsor, it's very important to make sure you have a lab manual that is easily understandable. If you want sites to conduct research in the proper way, you need to make sure that you're giving them resources that can be easily followed. And also just making sure that all of the labs that we're doing are not repetitive. Of course, every visit, you're going to have certain labs you have to repeat, but within the actual visit, that's not repetitive. Decreasing the amount of tubes that we're drawing, that's very important. If we can get 5 results from that one tube, let's do that. There's no reason for us to draw 26 tubes.
Oh my gosh, exploratory endpoints can absolutely destroy a trial. So many trials I've seen, there's 5 ICFs that can be signed. The optional ICFs, the
Yes.
genetic markers, the biomarkers, all this other stuff. Every single time you add something like that, you're adding additional patient suffering, burden on the site to ship things. Maybe for the sponsors out there, I get it. I know your scientists want some extra data to play with, but is this really relevant for the trial? Is this
Yeah.
really something that we want to push out to the sites? Because it could actually compromise our primary objectives.
Yes, it might be a hot take. Many people may not agree with me on this, but I believe that we should have more primary and backup aliquots. You should have primary, secondary, or backups, or whatever you may want to call it. You don't want to have to call the patient in to come in for a redraw. But also, what if it was a time point? What if it was a PK sample? You can't get that timing back. It is important that you save those samples, that you are not constantly harming the patient by doing another unnecessary blood draw. They're so important. I feel like every study should have it.
And for all of you sites out there, we've seen just recently wildfires, floods, tornadoes, earthquakes. Does your site have an emergency response protocol? And does that protocol protect your samples in the event your freezer goes out? Do you have a backup plan? How do you protect your samples? It's very scary to me, and I'm sure you've seen this with the sites you've worked with. How many sites will say, unfortunately, we don't have that?
Yes, so many sites. And then you have to talk out a plan and discuss what is the site capable of doing to handle that situation.
If I were to hand you the microphone right now and I said, what would you like to tell the entire industry about labs, lab processes, and what you'd like to see to make things better? What would you tell them?
Please take labs seriously. A lot of people end up taking labs as a small part of research. They may be a small part of your day, but they are a big reason as to why drugs get approved. We need to make sure that we're reading through those lab manuals. We need to make sure that we're prepared for each and every visit beforehand. We don't want to be putting out fires during the visit or while processing. We want to be well prepared. Just keep that in mind.
Take your labs seriously, respect them, understand why we're doing them and why sponsors are spending so much money to ship these blood samples all the way across the country to analyze. And if we can communicate with each other, plan appropriately, and always be prepared, and importantly respect our patients and the samples that they're giving, then maybe we can align our values and our principles in clinical research to the efficiency and throughput that we need for future generations and trial performance.
Yes, definitely. You have a way with words, Dr. Fox.
Well, thank you so much. Preeti, thank you so much for coming on to CRPN Central. An excellent conversation. I look forward to the live event.
Thank you so much, Dr. Fox.
I'll talk to you later. Bye.
Bye-bye.
Shanna has worked in lab management, operations, and logistics for years. Now she is the founder of VAMP, a mobile phlebotomy service that will collect every drop of a sample that a trial needs. It's a family thing. Labs are commonly the last thing that the site is worried about, although in reality, they are the critical data that our trials will need to be successful. If you don't know what you need, you don't know what you are missing. And as a result, sites should at all times treat the protocol and the lab manual like their Bible, ensuring they are familiar and aware of each trial's needs at all times. Sites should always pre-plan, plan, and double-check their preparations ahead of time with run-throughs before they scale into high enrollment numbers. And overly complex protocols and a lack of standardization are significant challenges to the industry. We need to focus on site burden to ensure our CRCs are not just collecting samples to collect, but rather focus on proper quality methods and accuracy. Ultimately, we are here for the patient, and if we respect the importance of labs, prepare constantly, and maintain thorough documentation and quality standards, our industry will eliminate missing data points, reduce unnecessary waste, and improve overall quality of lab results and trial performance. Preeti fell in love with clinical research and the zen of properly organized lab processes and procedures throughout her physician career. Now, as co-founder of Cornerstone, she brings her expertise and her passion for organization to every one of her site clients. Before a patient walks into the door, Preeti thoroughly reviews and understands the protocol and lab manual. She makes her own notes and performs mock lab processes to ensure she has all of the supplies, steps, and time that she needs to properly process patient samples. And she recommends sites plan everything about lab performance well ahead of time, from lab kit inspections and backups to supplies, labels, and scheduling shipping pickups. We must maintain thorough documentation to protect our sites after samples have shipped and they are in transit, just in case the delivery truck explodes. And if we prepare and plan continuously, eliminate unnecessary exploratory endpoints and inefficient low-quality processes, and respect our samples as much as we respect our patients, we may improve lab data and the industry's overall experience for our stakeholders. Both of today's guests confirmed the underlying importance of labs and their results in clinical trials, and both unanimously confirmed the importance of planning and preparation throughout the lab management process, and the importance of clear and easy to understand protocols. And while Shanna and VAMP are dedicated to reducing the burdens that our CRCs must carry and improving quality lab processing, Preeti highlights the importance of primary and secondary samples to eliminate sample redraws and unnecessary patient discomfort. So, how may we improve our lab practices for fast, efficient, and productive clinical trial performance? Shanna's third-generation phlebotomy legacy and Preeti's extensive lab processing resonates into three distinct best practices. To improve our industry's lab samples and results, we must focus closely on early preparation, clear communication, and thorough documentation. Plan early and often by understanding the protocol and ensuring you have the proper supplies. Create protocols and lab manuals that are clear and simple to follow, and adopt the detailed documentation practices that we need to protect our sites, our samples, and the integrity of our data for successful lab results and clinical trial performance throughout the industry. Thank you for listening and learning today about labs in the industry. As common as they are, it's amazing how very important and sensitive our lab processes are, and how critical their results are to the overall success of our trials. If you have any questions about labs or would like to follow up with some of the best professionals in the industry, reach out to VAMP or Cornerstone to see how they may help your organization with its labs and quality needs. Thank you to Shanna and Preeti for taking the time to speak with us today. Thank you to our CRPN Short Voice volunteers for starting the conversation. Finally, thank you to my sponsors for making this podcast possible. Thank you again for listening, and we'll see you next time on CRPN Central.